So in this video, I want to talk about the receptors that are needed to initiate an innate immune response. So these receptors are commonly referred as pattern recognition receptors because they recognize patterns on pathogens. So they can only recognize classes of pathogens, and they're not very specific. So generally, we distinguish between activating PRRs and phagocytic PRRs. Let's start talking about the phagocytic PRRs. They're very straightforward. What they just do is enabling phagocytosis. So for example, a macrophage is going to recognize that a bacteria has now invaded any tissue and it's going to initiate phagocytosis. And there's, for example, a mannose receptor that just recognizes mannose on a bacteria and therefore initiates the phagocytotic process. We also have so-called activating PRRs, and that's what you're going to hear a lot about, like TLRs, toll-like receptors, NLRs, knot-like receptors, RLRs, rig-like receptors, and they activate cell-specific innate immune responses. So they activate whatever this cell is supposed to do now in the innate immune response. So as we know, the macrophage needs to start producing pro-inflammatory cytokines in order to set the stage that other cells can help clear the infection. The dendritic cell is our cell that's going to run off to the lymph node to initiate the adaptive response. Therefore, the activation via PRRs will help to upregulate co-stimulatory molecules like B7, upregulate MHC class 1, MHC class 2. The neutrophil will be just activated to kill better, and epithelial cells that have RLRs, and when they're infected with a virus, they start also producing cytokines like type 1 interferon. So in order to know which kind of infection is going to activate which pattern recognition receptor, we need to know where they are located. So location is here crucial because that's going to tell us which invader is going to activate which PRR. The TLRs come in two flavors. We find them on the surface, extracellular TLRs, but we find them also in the endosome. So these are the endosomal TLRs. And then the NLRs and the RLRs we find in the cytosol. And so obviously, extracellular TLRs can recognize structures on the surface of a pathogen. In contrast, the endosomal TLRs, NLRs, and RLRs are going to recognize stuff that has, for example, been digested already in the lysosomes or that lives in the cytosol. So that's going to be most likely microbial nucleic acids. So if we know the location, we can also predict which receptors are going to be activated when we are infected with a specific pathogen. So when we are, for example, dealing with extracellular bacteria, you can imagine that obviously the extracellular TLR will pick these extracellular bacteria up, but also there's going to be always something phagocytosed, so it's going to end up in the endosome, and the endosomal TLR is going to recognize that. But in the cytosol, we're not going to find extracellular bacteria, so no activation of NLRs and RLRs. Then we have also bacteria that live inside the lysosome. So obviously, first, they are also going to be recognized by extracellular bacteria when they're just floating by, but once they're phagocytosed and are going to show up in the endosome, we're also going to recognize them by endosomal TLRs. And again, they don't normally show up in the cytosol, so no NLR RLR recognition. The viruses are going to be recognized by anything because they can float around and are going to recognize by the extracellular TLR. They're going to be phagocytosed and they're going to show up in the endosome, are going to be recognized by the endosomal TLR. But some viruses also bud out of the endosome and then release their DNA or RNA into the host cell cytosol. So definitively then they can be recognized by NLRs and RLRs because they normally use the host machinery to replicate. Certainly not all cells are going to be infected by the virus. So we can also distinguish which receptors are going to be activated in an infected versus an uninfected cell. So let's just take an example and say a virus that does not infect a cell. Well, it can be definitively recognized by the extracellular TLR when it's floating around, or when it's taken up and phagocytosed, it can be recognized by the endosomal TLR. But then, if it's not infecting and it's not going to use the host machinery to replicate, it would not be recognized by NLRs and RLRs. However, if you're dealing with an infected cell, so then it's going to bud out, release the RNA or DNA, and then use the host machinery. Then this would be an infected cell, and then the NLRs and RLRs are certainly also going to recognize and respond to it. So I just want to finish up with a general response that you can expect to the certain PRR. So if TLRs are activated, they normally give us pro-inflammatory cytokines like TNF, IL-1, IL-12. If NLRs are activated, we get predominantly IL-1, but IL-1 can then also stimulate other pro-inflammatory cytokines like TNF, IL-12, IL-6. RLRs generally give us type 1 interferon. This concludes the video on pattern recognition receptors.